Hello, my fellow scientists. I'm Peter Allen, and I will be joining you this year for a daily vlog on the topical science of the day. And that's going to include some of my own research as well as things that I find really interesting from what I've been reading of scientific progress. So in terms of my research, I'll be talking about some of the DNA nanotechnology that we've been developing here in my lab at the University of Idaho, as well as some of the applications to which we'd like to put it, including rapid and point-of-care diagnostics. What do I mean by DNA nanotechnology? What I mean is that DNA is not just the molecule that carries genetic information, it's also just a molecule, and we know how to synthesize that molecule. We can make DNA with basically whatever sequence we want. This isn't DNA from any particular organism, and it isn't going into an organism. It's just the molecule we want to use, and there are rules for how DNA molecules fold up, and there are rules on how DNA molecules react with one another, and we can use those rules to our advantage to do some really interesting things, including measuring biochemistry rapidly and efficiently. So my hope is to take DNA nanotechnology and use it as the guts of a rapid point-of-care test, like a pregnancy test, but for more sophisticated problems. As an example, there's a really difficult diagnostic for a disease called fibromyalgia, and this diagnostic requires that white blood cells be harvested, stimulated, cultured, and then the culture media needs to be tested for certain biochemistry, and that's a really long and difficult process, and there are tricks to making it faster. But ultimately, a lot of the time and effort comes down to processing those biological samples. What would be really awesome is if you had a dipstick that you could take the blood, culture it for half an hour, and then dip this stick in and read out a positive, negative for fibromyalgia. That'd be a lot faster than sending it to a lab and waiting 2 weeks in pain while your doctor tries to figure out if you're a likely candidate for fibromyalgia treatment. So I think this is a quality of life issue, and I think that my lab is poised to make some progress in that, and I hope to present results here as we get them in and as we publish them this year. In terms of the wider scope of the research that I'm doing and that I'm interested in, I think that the open source movement is moving into science, or maybe science is finally moving into the open source movement, and I think that's amazing and wonderful that there is now open hardware, open source scientific hardware, open source scientific instruments that can make research faster, cheaper, and more accessible to everyone, as well as making it more repeatable as different groups can build the instruments to repeat each other's work. Science is hard enough without having to spend $100,000 to buy an instrument just so you can repeat someone else's work. And I think that in terms of the quality of the scientific enterprise, it's dangerous to have these dollar-based firewalls around people's work, right? If it costs $1 million to repeat, how can we repeat? Is it even worth the funding agency's dollars to give another $1 million to someone just to repeat something that's come before? The answer has been no. We generally don't fund repeated work. They don't publish repeated work, so it doesn't happen, and that's to science's detriment. And I think that open source scientific hardware can help that. So I'm going to talk more about that tomorrow. I hope you'll join me. Thank you very much